Thanks for joining us in this session. We will try to make it very fast and very easy to digest session. So I don't know if you've heard of this before, but I will just give some overview on the Great Gene Reference Catalog initiative and how this will be managed later on within the Grapedia portal. So I'll give a little bit of examples of the two different resources and how all this is integrated within Grapedia. So first of all, I would like to thank my team who has been helping in the organization and the preparation of the workshop, especially David, Luis, Chen, and Antonio. Because we are just a few people today, we're gonna be all together in the practical session. This morning we were divided in smaller groups, but I think that we can easily work together today in the practical session. So first we're gonna explain a little bit more the theory, and I'm in charge of explaining the logic behind the catalog and how this is inserted in Grapedia portal, and then David is gonna explain a little bit more in detail the different parts of the submission form. So just for you to know, we have a lot of interest in the whole Grapevine community. You can see here all the guests that have registered in either the session of today or Monday session. So we were very happy to see that basically all the Grapevine community was going to be present in this workshop. The idea is that people learn how to use the catalog and learn how to submit genes of interest and their functions related to the literature in the database, because at the end of the day, what we want is to standardize the names of genes and the IDs that we are using. So in the future, if I have a gene, I want to know if that gene has been already named by somebody, and I would like to have a resource. We would like to have a resource as a community where we can put all together the functional information data regarding these genes. So I presume that most of you that are present today are interested in gene characterization, whether it's to simply understand the mechanism in which a gene is involved, or because you have an idea on an application on how to use this gene for the improvement of Grapevine. For all of us, it's very important to have a unified system for all the literature data that we can find for grape genes, right? Grape genes can be identified before we characterize them through different approaches. So I here just enumerate some of them. You can find genes that are interesting because you find them in genetic resources. So in a cultivar that has a different expression pattern, or that it has a different sequence compared to the reference. Also because it's found different in sequence or expression in a somatic variant, or even through QTLs or GWAS analysis, right? This transcriptomic analysis can lead to a reduced number of genes that have a particular interest in expression behavior, and we want to analyze one of these genes, right? And in other cases, it could be also derived from the inference of another species, of an ortholog gene in another species. So if you're interested in a gene that is involved in anthocyanin production, or that is related to fruit development, and you know that that gene has a function in tomato or in strawberry or in another crop, you would like probably to look for that same gene in grape and characterize it, right? So the sources of gene identifications of gene of interests is quite broad. When we do gene functional characterization, we can apply different approaches, and these approaches are somehow reflected in the submission form that you will learn how to use. So the data that we have gathered so far and the data that we want you to help us include in the catalog will be related to a certain level of validation of the function of that gene based on the experiments and the approaches, the experimental approaches or computational approaches that researchers or you, in the case you have characterized the gene, have used for that particular experiment. These genes are then used in different approaches and for the improvement of grape, and nowadays we have technology and the methodology to improve grapes using cisgenics or CRISPR-Cas by editing genes, but also it could be simply by using these genes and their sequences for breeding programs, right, in market-assisted selection programs. At the end, what we expect is to have an improved or a novel genotype, or to understand a mechanism under which a gene has its role, right? So back to 3 years ago, we started to see or to realize that it was very important to have a unified resource for gene functions. You can see here that the publications regarding or related to genes roles or functions, genes that are being characterized, are articles that are related to gene families, have been growing exponentially since the first release of the genome assembly, of the PN424 genome assembly, which was in 2007. Nevertheless, even in the early 90s, we had already some articles describing gene functions in grapevine. And right now, we have all of these genes, but there were no resource, no gene function database or literature database to have all this information, right? So that was a first need. Of course, we can use different databases as a model for us to try to develop a similar database. The best example is the Arabidopsis information resource that probably some of you may have used in the past or in the present. This database, it's quite nice because it links gene information that is found in the literature with other resources, for instance, related to gene expression, to gene ontologies, to gene phylogenetic analysis, to find orthologs in other species, or even to a collection of tDNA insertion mutants. Unfortunately, we don't have tDNA insertion mutants collections for grape, but we do have somatic variants. So why not having a resource where we can have genes associated to somatic variant phenotypes, QTL databases, and so on, right? So we started to work more than a year ago on a database that it's an open source database that is called Grapedia, which stands for the Grapevine Genomics Encyclopedia. This database, it's basically a modular system composed of a federative database system, and that will have in different modules different types of information. So we expect to have a module related to transcriptomes, a module related to genomes, and a module related to gene function data, so literature-related data, right? The idea is that these modules are interoperating, and you will be able to jump from one module to the other in one single user interface. The three main pillars of the Grapedia database are first the community, so all the resources that are generated by the community that are free available and that we will offer them also in open access within the portal. And these are very exhaustive. We have different types of data. We have omics data, we have phenotypes data, we have literature data. And the idea will be to implement and integrate all of this data that is quite exhaustive. And in the past 30 years, the Grapevine community has been generated a lot of this data, especially in the last 2 decades. Then all these data will be integrated, or it's already being integrated in Grapedia with this modular architecture. And then the third important pillar is what we will offer through the database, right? So we will offer different services that are, most of them open access, are free to use, that are consistent basically in browsers, dashboards, and workflows. For instance, we will have a particular function of the gene catalog, which is called the gene cards, which I will explain later on, as part of a dashboard. So we will be able to jump into the gene card of a gene, but from there we can move to another place, for instance, to analyze the SNPs that we find in different genomes for that particular gene, or the expression that we find for that particular gene, or the co-expression, and so on. Most of these analyses and also some workflows, which are basically analyses that you will be able to run inside the portal or download and run it locally, most of these services are going to be free and open access. However, we have designed some customized possibilities, some customized services that will be offered to the community in case they want to use these services, and we will apply a fee for those because we want to maintain the database in the long-term basis. These fees will be able to help us maintain the database and allow the sustainability in the long term. Very briefly, the Grapedia portal has two basic structures. One is the back end, which allocates all the data. This data can be located in one or more different places. That's the whole idea of a federative data system, actually. So the data will be extracted and it will be interconnected, and we will be able to query and explore this data that will be in the backend through different components, reactive components that will be in the front end. The front end is basically what we will see in the user interface. And this will be the web, the dashboard, and the workflows. So what we want you to help us within the Grape Catalog initiative is to give us the data that we need to include in the backend, and we will offer it through the front end so you can explore and use it in the best way for your research. The Grape Gene Reference Catalog was already established a couple of years ago, on 2021, and since then we have already a second version. These two first versions of the Gene Catalog have been based on this scheme in which we have two different interfaces. One is the researcher interface and the other is the catalog curator interface. Initially, a group of manual curators were searching for all the published data regarding gene functions or gene families, and that was included in the database, given a validation level that I will explain later on, and associated those genes to the same genes in other genomes. For instance, different versions of the same reference genome, or other cultivars or other species. And that is something that we are now including. So in the case that we have genes that are not present in the reference, but are present in other genomes, let's say wild BTs or American BTs or different cultivars, we will be able to search for those genes. All of this information, the function, the literature related data, and also the correspondences between all the different genome assemblies is being incorporated in the catalog. So the catalog was originally available in the Integrate website, but now it's available in the Grapedia website. So you can go in our website and explore the catalog. The catalog itself has conversions, have function and validation levels, has also the membership of the genes to different families and pathways, and of course has the links to all the literature. One more application of this catalog that we have generated it's what is called the Gene Cards app. So it's what you see on the right side of this image. We have been able to download all the public transcriptomic data from the Sequence Read Archive, SRA, in NCBI, and we have reprocessed the data and generated expression matrices that we offer them as dynamic and a very nice tool to navigate through the genes and see the level of expression of these genes in different organs. I'll show you some examples later on. The Gene Cards app is present now in the BitBees platform, but it's already being incorporated also in the Grapedia portal. The catalog data, the expression data, and even future links to gene co-expression networks will be available within the Gene Cards app. Both the catalog and the Gene Cards have the possibility to be downloaded, whether they are CSV or Excel files, or even annotation files that you could eventually use for your transcriptome analysis, already have the gene IDs of the catalog. So when you are doing a transcriptome analysis and you run down the differential expression analysis, you can already have the gene name that is given within the catalog. Okay, so the catalog included level of validation for the genes that are included in these results. This level of validation depends on the degree of the characterization. So we would have at the beginning to do a manual inspection of all the experiments that were conducted for these first genes. The idea is that the community, now that it has been established, will include their own genes that they are working with, and they give the experimental validation according to these criteria. So the experimental validation, it's a scale from 1 to 6, in which we have hypothetical genes of the first validation, the lowest validation level, which are simply genes that have been identified as a putative, no, in this case, as an hypothetical function based on similarity to other proteins in other databases. And in this case, by using different search tools such as BLAST, hidden Markov models, or even perform motif search, right? The level 2 would be what we call putative functions, which include genes that have been identified in the whole family, gene family context. So imagine that we are interested in a transcription factor. If we have an article that describes the whole family, and we have all the genes that belong to this family, and this includes phylogenetic trees and some expression data, we will consider this validation of these roles as a second level of validation. We also have the third, which is the proposed one, in which we have correlation experiments to give or to propose functions for these genes. And these, for instance, are gene co-expression networks or correlation to metabolite profiles. So if we have an article in which a gene has been proposed as related to hormones, because it's highly correlated to a particular hormone profile, we can give it a validation level 3. Finally, in the level 4, we have candidate genes that have been associated with QTL or GWAS studies. We, of course, know that QTLs are quite big and they contain hundreds of thousands of genes. So genes that have this level of validations are genes that have been identified through QTL mapping, for instance, but have been suggested in the article as candidate gene based on expression behavior or sequence, or simply because of the nature of the gene. Finally, we have the two highest levels of validation, which is 5 and 6 for genes that have been validated based on molecular studies, such as knockout, overexpression, or negative dominance. And the differences between these two levels vary depending on the species in which the validation was conducted. So level 5 would be genes of grape that have been validated by overexpressing them, for instance, in a heterologous species. For instance, anthocyanin pathway gene that has been overexpressed in tobacco, and you see anthocyanins produced, and the gene has validated role in the production of anthocyanin, but it has been done in a different species, not in beeties. So that would be given a validation level 5. Validation level 6 includes genes that have been validated using knockout or loss of function, silencing, or overexpression in the same species. And we have also included in this level of validation genes such as those coding for enzymes that have been characterized using in vitro or recombinant methodologies. So we consider that that is also the highest way to demonstrate the role of an enzyme. So by 2021, we had incorporated 1,000 genes in the catalog, and you can explore them, not only the functions, but also their expressions. And by 2022, we had double number of genes. We expect by the end of this workshop, around November 2023, to have the third version. And hopefully all the people that are joining us in this workshop and in the other sessions will include many more genes. So one of the things that we are also very much interested is in the correspondency of genes between different genome versions and different genome assemblies and different cultivars of different species. So in the case of the PN424 reference cultivar, you know that we have, in the past 2 decades, several assembly initiatives and also several annotation versions. It has been a quite complex journey of different naming systems, different IDs, but you have to consider that each one of these different assemblies has, in some of the cases, consider a completely new annotation pipeline, automatic annotation pipeline. So it is somehow difficult to have a full correspondency between different annotations. Well, we have done this already. So we have been able to assert the correspondency between different annotations for a same gene. And while you submit your genes in the submission form, you will be able to include the gene ID of the gene regarding the ID that was used in the article where this gene was published. Okay, so that is very important. For submitting the genes, and this is something that we will go on later on, to submit a gene in the catalog, we need a publication, so you need to have the access to that paper or that article, or at least you should have the information of that gene in the case if you are characterizing that gene. This is an example of how the gene card looks. So for instance, we have a gene here that is the pectin metylesterase inhibitor that is not currently present in the catalog, but we still have expression data for this gene. So as you can see here, this gene is not present, but the idea would be to include all of these genes in the catalog. And finally, we are very much interested in identifying gene sets between different cultivars and between different Bt species. We know now that recent studies have characterized some pangenomes in Bts, in the Bts genus. So we know that by comparing these genomes and putting them together in a pangenome, we have a very high number of genes as we increase the number of genomes in this pangenome. And because this may give a lot of interesting traits, and these could be genes that are actually responsible of resistant traits or quality traits, we need to consider them as well. So we cannot stay only in the genes that are present in the reference. We need to go beyond and look for genes that are not only present in the reference, but are also present in other genomes, or that are exclusively present in other genomes. Whether our pangenomes are closed or open regarding the percentage or the overlap of genes that are shared between these different genomes. No matter it's one or the other, the Beatus pangenome is already telling us there are hundreds of thousands of genes that need to be included in the catalog. So before I end, I will invite you to use the Grapedia website. You know that we have a lot of resources already in the Grapedia. The database itself, it's in construction. We have developed a pilot that is already being tested, and we hope to have an alpha testing model by the early months of next year. And however, you can still look for genomes, explore genomes, and also the genes in the Gene Catalog and the Gene Cards app. And also in the Grapedia portal, you will have the submission form, a form example, and the submission guidelines. So once you finish this workshop, in addition to what you have learned from us, you will also have this documentation for you to continue learning and continue submitting genes. And most importantly, we hope that you will be able to transfer the information to your partners. So with that said, I want to thank all of you again for being today in this session, and I will leave the stage to David so he can present his part. Thanks, everyone.
Okay, I'll share my screen now. Thank you, Tom. Okay. Can you see my presentation all right?
Yes, everything all right.
Yes.
Perfect. Okay, so I think from here and maybe like start a bit again with the basics, but the main thing that the catalog is centered on is the idea of a gene, which is still a useful one, right? Even if a gene like may encode for different proteins, like and have different functions in a way, we still like in biology try to think of like the functional unit being the gene, and that's how publications work at the moment, and it's certainly in other species and also in Beatus, right? So you can think of like the gene being like the biological concept, and then the gene model more like something that changes depending on your map of the genome, so your genome assembly and the annotation. And this is something that is reflected by the gene IDs. As you see in Grapevine, we've got many different annotation versions just with one genome. We'll soon have even more genomes and more IDs, so this is something we have to get used to. And then the gene symbol is mostly absent, actually, like Tom mentioned before the catalog, we didn't have a resource where we could find this, and researchers name their genes, but often there are many cases of renaming genes that were already named in the past, just because there's not a single resource to refer to. So we're moving towards a situation where you can follow your gene through the gene symbol, but also be aware that different assemblies may reflect a different nature of your gene or slight differences. So here's an example that you can see, a typical example in the reference genome where the model can change quite a bit, even though it's still referring to the same gene. Like, for example, Lacase 40, we can have different models, and that's why it's important also to reflect the gene ID that the work describing the gene came from. So yeah, going back to this, there are two main aims for the catalog, right? Describing the function of a gene and provide the level of validation, as Tom was mentioning before. Cite the literature appropriately so that it's open for everybody to see and compare. And also have these unique gene symbols that are associated to gene IDs from, well, ideally the current annotation version that we've got in PN. I will explain later what we're proposing in this sense. And also those genes that are only present in other cultivars or species can also be added, or we're opening the possibility. So they will have IDs from another assembly, but none in the reference genome, of course. So going to the functional validation score, as Tom explained well, we've got different levels like that will support your function that you describe in one of the columns, and it ranges from a simple hypothetical case where you've got a sequence-based homology to a protein in another species and you're suggesting a function, to actual knockout and loss of function experiments in VTC itself that are the closest or the highest level of evidence we're contemplating at the moment. In the past, the previous versions of the catalog have been based on V3 gene IDs. So the latest annotation of the 12xv2 genome, and the gene symbols, we've got around like 2,000 gene symbols so far. And the gene ID conversions were based on previous correspondences that were done, but these were not exhaustive. Like we had conversions to V1, V0, and NCBI, but they didn't contemplate, for example, V4 or 8X. So in terms of like going through the literature, you still have some studies done in 8, like at the beginning they were done in 8X that could not be easily incorporated into the catalog. So this is something that we have fixed, and now the user can just use the IDs that you find in the publication rather than converting them yourselves. We'll do this behind the scene, basically. So this is the general process where you can go and access the Grapedia website, and there you will find, as you saw before, the submission forms, example forms that are already filled in, the catalog guidelines, which are supposed to help you. Like they should be a standalone guide, but yeah, so you can share it with other people that haven't attended the session, and they may also submit genes to the website. At the moment, we are receiving the forms by email, but we'll also be open the possibility to upload them on the website. But the main idea is to end up with a catalog before the end of this year, where all the submissions, the recent submissions, are incorporated. And we've added to the submission forms a few columns with respect to the past, right? So since now we're opening the possibility to upload genes from other cultivars, we've added these respective columns. The main thing to notice is that although this is a possibility, most of you will be adding genes that are associated to the reference cultivar PN4024, because most researchers, even if they're working with different cultivars, they will provide accessions that correspond to their reference. So in that case, you will find yourself mostly using these genes. It's just that we're opening the possibility to do otherwise. And in the part of the functional description, we've added some columns that are regarding the species and cultivars, just because we often find the case where a gene has been studied in many different cultivars, and you may find that the function varies slightly, or in any case, it could vary. So what we're trying to do is like to annotate the function, write down the function of the gene, but also with respect to the cultivar that corresponds to the work. So let's say MIP15 has been tested in Merlot for like overexpression, and then silence in another cultivar, and something else was seen. So this is something we want to note down, since the differences between cultivars is often what makes researchers be interested in a gene. We've kept the input as flexible as possible. Like as I mentioned before, we're now allowing any gene ID to be submitted from the reference genome, besides those rare cases we mentioned of different cultivar IDs. And then this means that this information will then be split or distributed across the different resources. So some information will go to the gene catalog, the table, some will be reserved for the gene cards, and all the information will be available in Grapedia and the other tools for visualization that we have in BitVis, that will allow you to track your gene with the symbols that you have provided. It will be more user-friendly like this. And so the main idea here is that the gene catalog, even though we're allowing gene symbols that are missing from the reference, the catalog will only have IDs for the reference genome, just because we cannot have more than 20 or even like in the future, maybe hundreds of columns like corresponding to each genome annexation. So the table will be kept simple, like with one ID column with NAs for those cases that are missing, but the information regarding IDs in different cultivars will be shifted to gene cards. So we're not losing that. So that's why the submission form picks up that information. So the correspondences between the reference genome IDs have already been worked out, although we are still working towards correspondences between different cultivars, since the same method cannot be used, as the genome differences between cultivars is obviously much higher than what we find between assembly updates. Okay, the final catalog table will look a bit like this, where with these columns being the ones that change with respect to that may change with respect to the submitted form, right? Because we'll update the gene IDs to the latest PN4024 annotation, and that may involve checking if the gene is now duplicated in the newest assembly. So that might mean that the gene symbol is also modified. Let's say MIB15, we find an extra copy of MIB15, then the gene symbol would be MIB15A and MIB15B. This sort of thing will be done automatically, but you still, but we'll still keep trace of like the original ID submitted, as this will give us traceability to the published study. And if there are any errors, like in converting this gene ID to the new assembly, we'll be able to fix them since we're dealing with them as well. The same thing, if we're changing gene names, they will go to the gene synonym, in the sense that we'll also keep like a traceability of all the names that a gene had in the past. And some of the information that is will be kept by gene cards is mainly the equivalences to other PN4024 annotations, and also further genomes and assemblies. When we update the catalog this time around, we're going to wait for the new T2T annotation, since it's been recently worked on, and it should be like the best annotation so far for a grapevine genome. So we're showing you here like what the ID will look like, because we think that it's a good idea to have an ID that is recognizable, or that it can be pinpointed to a particular assembly, but still the structure of the ID doesn't change that much every time we go into a new annotation version or even a new genome version. But we do want the ID to reflect what assembly it came from. This is especially important now, when people may even do experiments based on the synthesis of a gene derived from the annotation sequence. So the small differences that we may have in a gene model, although they still refer to the same gene, will matter biologically in some cases and papers. So it's important that we make the ID reflect that time, that genome or assembly that is being used at that time. Okay, so this is an example of why it's useful to add your genes to the catalog, in that you'll be able to search for them in the different applications we've got, that will give you access to a lot of transcriptomic data, for example, that will give you an idea of where your gene is expressed and in what kind of conditions, right? So it's something that will be in your interest, as well as the community. We are also trying to implement the BitVis apps that we've got in Grapedia, and there will also be like further information linking to other genomes, as we already mentioned. This is a bit, this is an overview of what the submission form looks like. We try to make it as comprehensive as possible, in the sense that it helps you like fill it as you go along, with showing you which fields are compulsory, and together with the guidelines, which you can download from the Grapedia website, or you can even click this link and download them again, so that you always have the latest forms and annotation and submission guides. You should be able to fill genes appropriately, but I think since some cases are maybe complex, like in the sense that you may want to replace a gene, or you don't know if you're sure that your gene has to be added to the catalog, we've provided a simple enough decision chart that will guide you through that process. Like, for example, you first start asking if your gene of interest is already in the catalog. So this would involve downloading the current version of the catalog and searching for your gene ID or gene symbol and seeing if it is there. Of course, like the catalog is only showing the gene IDs of the current version. So if you need to find a conversion yourself, you can go to the VT's visualization platform that will keep the conversions, or Grapedia soon enough, and you will be able to check for sure that your gene ID is missing from the catalog. If it is missing, then you need to ask yourself if the cultivar the gene from the cultivar that you're working in has an ortholog in PN4024, which most of the cases the answer will be yes. And in those cases, you need to question if the study of the gene function is already published. If it's not published, we recommend to use the latest PN4024 gene IDs and annotation, and prepare the document for publication like this. Once it's prepared and maybe you're almost ready to publish, or like you have a bioarchive, then yes, you are ready to submit. But the idea is that submissions go together with a paper, let's say, that we can refer to. And if the study is already published, we recommend to submit the IDs as reported by the study, because like that we have a traceability. Even though in the final catalog you'll have the updated annotation version, we'll keep the original ID to trace potential errors if we find them. In the case that your gene doesn't have an ortholog in the reference cultivar, we need to ask the question of, does the cultivar species have a published genome assembly or annotation? If it has one, then you can submit your gene name, your new gene name, using the accession-specific gene IDs. So whatever annotation format they use in your cultivar of interest. If it doesn't have an assembly and annotation, but you're still sure there's no ortholog of your gene, maybe you have phenotypic evidence, or through BLAST, the highest hit is still like under 80% identity at the CDS level, for example. So some kind of evidence that really supports that your gene is not in the reference, then you can submit an entry without an ID, as long as the functional evidence for that gene is above, like, let's say, a value of 3. So quite solid evidence to justify giving a name to something that you cannot assign a sequence to, let's say, from any assembly. Okay, and then maybe less of the cases, but also important, are those where the gene is already in the catalog, but you want to add information, or even correct or replace some of it because you think it's incorrect. In those cases, these submissions are going to be reviewed mostly manually. So it's very important that you choose the right submission type. You'll see later in the form, and also that you provide careful comments in what you propose to do. Let's say you may just want to add information to the entry and not change the gene symbol or gene name, or you're maybe proposing to change the gene symbol. But then in those cases, you need to argue a bit why, because we want to know, I mean, let's say you think like that this other reference is more appropriate because they've done a more careful study or a more comprehensive analysis of a gene family, and then we should go with those gene names instead. This sort of thing will also be accepting. So even if, or the most simple case where you just want to add another citation to another form of the function of that gene, that's no problem. Like that will be more easy to process it. You just just submit the genes as a add information mode, then we will just incorporate that information to the existing entry. Anyway, thank you all for listening.